Hola, ¿cómo están, my compañeros? I'm right here reading this very interesting article. It's called Want to Eradicate Viruses? They Made Us Who We Are. It was written in 2017. I'm going to spare you and skip down to the bottom. Something very concerning. I will start right here. Well, the virus particle may fall short of the definition of life, depending on the criteria used. For some virologists like myself, thinking of the viron as a virus is like calling a sperm or unfertilized egg a person. Sure, sperm is an essential step towards creating a person, but few people would argue that a sperm or unfertilized egg should be described as the finished product. Much like a sperm, virons are produced in the millions. Many will never reach their destination and are lost and degrade in the environment. It is only when the virus binds to and enters a target cell that its cycle of replication can begin. A viron does not even always contain a majority of the molecules a virus can create. For example, the norovirus viron contains just 3 different types of protein and 1 type of RNA, a nucleic acid like DNA, which uses a different sugar to form its backbone. Infected cells, however, make at least 8 different viral proteins and 4 different viral RNAs. Nor does the virus particle itself usually result in the symptoms of the disease. Typically, when you catch a virus, your symptoms come from either infected cells dying or your immune response to those infected cells. For these reasons, some virologists consider the infected cell rather than the viron to be the virus. That is saying right there that when a virus goes and hits your body, usually what happens is the same typical immune response. And it's saying that typical immune response initiates chemical signals, but it also initiates signs and symptoms, which are ubiquitous amongst most viruses. And for these reasons, some virologists consider the infected cell rather the viron to be the virus. So the infected cell is the virus, then the mRNA vaccine, which infects the cell, is the virus, creates the virus. That's what the mRNA vaccine does. It goes into your cell and enslaves your cell and forces your cell to produce a protein, which you have never produced before. And I'm going to continue reading. I am a virus. Well, this idea sounds outlandish. From conception to grave, your cells are intrinsically associated with viruses. Even if you don't have a cold or the flu, you are still part virus, as human DNA plays host to a range of different viruses. These are retroviruses, the best known example of which is HIV. Well, HIV only entered the human population relatively recently. Viruses very much like it have been infecting us and creatures we evolved from since long before humans existed. Well, HIV infects immune cells. When a retrovirus instead infects the cells that produce eggs or sperm, the viral DNA can be inherited by any offspring. Over 1 million years of these viruses have lost their ability to produce infectious particles, but have in some cases found other vital roles and are now indispensable for human life. Viruses inside of us that are indispensable for a human life. These viruses are indispensable for human life. And this is the last paragraph, is absolutely crucial, okay? One well-studied example is a protein called syncytin-1. I'm sorry, I can't pronounce it. One well-studied example is a protein called syncytin-1. This protein is created by a virus inside of your body. This protein is vital for the development of the placenta. This was originally a retroviral protein which entered the monkey population, which gave rise to humans around 24 million years ago. If we deleted this protein from our DNA, humanity would rapidly go extinct, as we can no longer produce a functional placenta. All these viruses, which inserted into our DNA long ago, are termed endogenous retroviruses. In humans, ERVs have long since lost the ability to produce infectious virons, but this is not the case in all animals. Pigs' ERVs, for example, can produce infectious particles and are concerned when considering the use of pig organs for transplant, as these are known to be able to infect human cells. Blurred lines. If a virus is the infected cell, rather than the viron, you could even think of the virus that can infect us as more than 99.9% human. This is because they need many of the human proteins or other molecules present in your cells and encoded in your DNA to make more virus. That's what the mRNA vaccine does. It goes into your body and it produces more particles. A human cell is vastly more complex than even the largest virus, and viruses can make use of this to compensate for their own simplicities. Viruses and their host cells share many common needs. They need to be able to produce RNA, protein, lipids, and have access to raw materials to generate these. As a host cell already contains all the needed components to achieve this, a virus can simply provide its own instructions in the form of the viral genome and let the cell do most of the work, which is exactly what the mRNA vaccine does. It enslaves the cell by cell. It's slavery at the cellular level. It takes many more cellular proteins to make a virus than it does viral proteins. A virus only needs to provide instructions for the few components the host cell cannot produce. An example of this would be a virus which has a viron with a lipid membrane such as influenza. This membrane is usually recycled from host cell membranes. The addition of a couple of viral proteins converts this into the membrane coat of the viron. That's meaning to say that it steals the bilipid layer of the host cell. It makes its little particles, and then when it extrudes itself from the cell, it steals its lipid layer. It doesn't have to be able to make a lipid layer, it just steals the lipid layer. This use of host components by viruses also makes it clear why it has been so difficult to develop effective antiviral drugs. Much as with cancer treatment, there's very little to distinguish infected cells from normal human cells, which makes coming up with a drug that will only target infected cells extremely challenging. To be effective, you have to target that tiny part of the infected cell that is purely virus without harming the remainder. So, are viruses alive? It's still not settled and really depends on what you think a virus is. What does seem clear, however, is that the virus which infect us can be seen as part of human. Viruses which infect us can be seen as part human, and we are part virus. I think this is an interesting article. One thing I wanted to note is the fact that there has been talk that this syncytin 1, which is vital for the development of the placenta, is actually what is under attack by the mRNA vaccine. I'm not 100% sure about this, but I have heard conspiracy rumors on the rumor mill. And for it to say right here, if we delete this protein from our DNA, humanity would rapidly go extinct as we can no longer produce a functional placenta. And that just right there, rapidly go extinct, that just hit me so hard when I heard that. It was like, wow, if they did that to us, if they could do that to us, they would do that to us. That would be us, that dopey pig waiting to get slaughtered.